Welcome to the Diabetes Bio podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we will interview one or more authors from the feature articles, the papers of the month, or other articles published in that month's issue of the Diabetes Journal. We also have a Sweet Talk segment of the podcast, which is more of an open format portion of the podcast that can include topical interviews or discussion. My name is Darlene Sandwell, and as always, I am joined by my podcast co-hosts, Dr. Kirk Habiger and Dr. Kevin Williams. How are y'all doing today?
Hey, good morning, everybody.
Morning, good morning.
So this weekend I hit my first, not to brag about living in Colorado or anything, but I hit my first above timberline hike for the summer with my good friend Fran and my faithful, as long as I'm taking her for a run or a hike, border collie named Jazz. It was a beautiful day with just a few fluffy clouds out in the distance and 360-degree views of the Rocky Mountains. So it was a great weekend. How about you all? What's your favorite summer activity?
Yeah, Darlene, first, I hate that for you. So my condolences. I think we're obligated to say ADA sessions, right? That has to be a summer activity that we look forward to. But if I'm talking about on the home front, we spend a lot of time outside. So some kayaking when it gets really warm, long runs in the morning before it gets really warm. I spent this weekend doing a lot of yard work, which was not hiking above the timberline. So yeah. How about you, Kevin?
Yeah, I mean, not to brag, a couple of weeks ago, I think we hit our first triple digit temperature day.
Wow.
Wasn't that fun? We went back down in temperature a little bit, but we're going to be back up there pretty soon. So, we got some doggos ourselves, so we like to get out and let them have their own little adventure. They definitely encourage that pretty much on a daily basis.
Yeah.
I do like to try and get out and go run. We also like to travel a bit, so maybe this summer we'll be able to explore some places that we haven't quite been to before, explore some new spots. Fingers crossed. Let's see if everything lines up.
Are the dogs
Good.
water dogs?
One of them is for sure, yeah. So trying to get them. We don't have a pool, so we can try and go to a lake, which is pretty close to us, and they definitely love that.
Yeah, cool. All right, well, I guess we should talk some science. Kirk, why don't you tell us about our interviews today?
June was a great month. We interviewed the paper of the month titled Hepatic PKA Mediates the Liver and Pancreatic Alpha Cell Crosstalk. We were joined by Kehan Bao and Haruka Okamoto from Regeneron Pharmaceuticals. It was a great discussion. And we also discussed this month a featured article, The Effect of Small Increases in Blood Glucose on Insulin Secretion and Endogenous Glucose Production in Humans. That discussion was with senior author Greg Kowalski, associate professor at Faculty of Health at Deakin University in Victoria, Australia.
Awesome. And Kevin, for our Sweet Talk segment, we have a kind of a sneak peek for one of our ADA Scientific Sessions award-winning lectures. Tell us about it.
That's right. We're speaking with Dr. Shingo Kajimura, who is this year's recipient of the Outstanding Scientific Achievement Award from the ADA. Dr. Kajimura really pioneered our understanding of adipose tissue development and biology, both in thermogenesis, but also in its broader role in metabolic health. I think it's really a fascinating conversation. We explore the trajectory of his career, his scientific insights, and the future of adipose biology in our understanding of metabolic disease. I think it's also important to remind our listeners, as Kirk kind of alluded to, for summer activities, that the 85th Annual ADA Scientific Sessions is coming up later this month, and there's still time to register. Over the past 2 months, we've interviewed the Banting Medal recipient, Dr. Stephen Kahn, and as I just mentioned, the Outstanding Scientific Achievement Award recipient, Dr. Shingo Kajimura. Check out these conversations in our podcast this month and last month, and then come see their lectures in person and much, much more.
All right, let's jump in.
Today, we're going to discuss June's paper of the month titled Hepatic PKA Mediates the Liver and Pancreatic Alpha Cell Crosstalk. We are joined by Drs. Kehan Bao and Haruka Okamoto from Regeneron Pharmaceuticals. Dr. Okamoto is a director in obesity research at Regeneron, and Dr. Bao is a postdoctoral fellow there. Welcome, Drs. Bao and Okamoto. Congratulations on your paper, and thank you for being part of this month's podcast.
Thank you for having us.
Thank you for inviting.
All right, we're going to throw a little bit of a wrench at you both and ask you an icebreaker question that we didn't provide ahead of time. So take your time if you want to think about it. But your question is, who is the most inspirational scientist to you, live or dead, and why? And we'll start with Dr. Okamoto.
To me, that would be Susumu Tonegawa because I read his book when I was in high school, and that inspired me to become a scientist.
Oh, that's great.
Wonderful.
Dr. Bao?
I would say maybe Dr. Sidney Brenner. I studied about him using C. elegans as a genetics tool to discover different gene functions, and that introduced me to the world of genetics. And since then, I've been in the genetics field ever since.
That's great.
Yeah, very good. So, Dr. Okamoto, as we get started, maybe you can give us an overview of your research program and then how this specific project fits into it.
Sure. So my lab has been studying glucagon signaling since mid-2000s, and our initial focus was to develop glucagon receptor inhibitors. The best described function of glucagon is to elevate blood glucose. So we hypothesized that the receptor blockade will lower glucose, and we indeed made an inhibitor that did so very effectively in animal models of type 2 diabetes. But what we also learned from the inhibitor was that blocking glucagon signaling causes some safety concerns, and one of them was an increase in the mass of pancreatic alpha cells, which produce glucagon. Since then, we've been studying the feedback loop between the liver, where the receptor is located, and alpha cells, and this paper describes our latest findings.
You know, Dar and I are both big fans of glucagon, so you're in a safe space here.
Yes, exactly. Let's talk a little bit more about that, the liver islet crosstalk. So maybe talk a little bit more about the history behind this and what you felt was the significant gap in knowledge that led to this particular project. Maybe what your central hypothesis was.
Sure. As Haruka mentioned, glucagon has long been known to regulate glucose production from the liver, but recently it is increasingly known as a potent regulator of amino acid metabolism. Activation of the glucagon receptor, GCGR, in the liver stimulates both the uptake and the breakdown of amino acids. So when the signaling pathway is inhibited in the liver, there is reduced amino acid breakdown in the liver, which can lead to an increase in the circulating amino acids. This is observed in GCGR knockout mice and also in human patients with GCGR mutations. What was also observed with these GCGR knockout mice is that the alpha cells, which are responsible for producing glucagon in the pancreas, they are hyperplasic. And several studies in the last decade, including studies from us, confirmed that the alpha cell hyperplasia seen with GCGR inhibition is directly caused by increased circulating amino acids. And since then, this phenomenon has been termed as the liver alpha cell axis. However, the question remains, what is the downstream pathway, downstream of GCGR, that mediates amino acid metabolism and alpha cell hyperplasia? And this is where we aim to answer in this paper.
Excellent.
Great. So continuing on that, this work dissects glucagon receptor signaling and downstream metabolic regulation, and you used AAV-mediated knockdown. Can you tell us a little bit about the targets that you chose for these studies, and also speak a little bit about the signaling intracellularly?
Sure. So GCGR is a G protein couple receptor. I couple is mainly through GS alpha, and downstream of GS alpha, there's adenyl cyclase activation and increase in cyclic AMP. And in response to increased cyclic AMP, there are two effectors described in the liver. One is the canonical protein kinase A, PKA pathway, and the other one is the EPAC2 pathway, which has also been described to have a role in different metabolic functions in the liver downstream of GCGR. So in this paper, we want to tease out which of these pathways regulate amino acid metabolism. So we use AAV to knock down GCGR, the G protein GS alpha, and the two effectors PKA and EPAC2.
Yeah, and there is some GQ coupling, right, to the glucagon receptor in the hepatocyte. Did you explore that pathway at all?
Yes, there is a GQ coupling. We focused on GS alpha because there was a paper about 20 years ago that liver-specific GS alpha knockout mice, they also develop alpha cell hyperplasia. So we narrowed down to GS alpha and focused on its downstream pathways.
Yeah, very good, very good.
So, can you tell us a little bit more about your findings and how you think the work that you've done here clarifies this link between amino acid metabolism and specifically the alpha cell hyperplasia?
Sure. So we knocked down PKA, EPAC2, GCGR, and GS alpha. What we found was that when you knock down GCGR, GS alpha, and PKA, there were increased circulating amino acids and alpha cell hyperplasia. Also in the liver, we saw suppression of genes involved in amino acid transport and catabolism. But EPAC2 knockdown, on the other hand, didn't have much an effect. So we confirmed that it is the PKA downstream of GCGR that mediates this liver alpha cell axis and not EPAC2. We saw alpha cell hyperplasia and increased circulating glucagon levels when we inhibited GCGR, GS alpha, and PKA. And we think the increased glucagon could come from two mechanisms. First, there is alpha cell hyperplasia. So the more alpha cells you have, probably more glucagon production. Secondly, there are some amino acids that are known to be stimulator of glucagon secretion, even in normal alpha cells. So we expect the increase in circulating amino acids can also directly stimulate glucagon secretion through those amino acids.
Did you look at any other pre-proglucagon peptides? So, GLP-1, for example.
There was a report that GCGR inhibition, there is an increase in GLP-1, and that is consistent with what we found as well.
Very good. You know, one of the experiments that really grabbed my attention from this paper involved glucagon receptor inhibition combined with PKA knockdown. Can you describe the findings of that study for us and what you interpret the outcome to mean?
Sure. From the knockdown study, we found that PKA seems to be the mediator of amino acid metabolism and alpha cell hyperplasia. So we say, let's try activate PKA and then see if we have the opposite effects. Also, we wanted to ask if PK activation is enough to reverse the effect of GCGR inhibition. So to this end, we activated PKA by knocking down PKA's major inhibitory subunits, PRKR1A. At the same time, we give these mice GCGR blocking antibodies. And what we found was that with GCGR inhibition alone, we saw increased circulating amino acids, alpha cell hyperplasia, as has been described before. And with PK activation alone, we observed the opposite effects, the reduced circulating amino acids and a reduced alpha cell mass and glucagon. Importantly, when we combine GCGR inhibition and PK activation, we have the same phenotype as PK activation alone. So this really says that if you activate PKA, you can fully reverse the effects of GCGR inhibition. I think this further supports our conclusion that PKA is the major mediator of amino acid catabolism and liver alpha cell axis in mouse.
Great.
So, Dr. Bao, you carefully measured multiple amino acids in this work. Can you provide some clarity on what specific amino acids might be triggering the hyperplasia in the alpha cell?
Sure. Many studies have been done on this area, including us. Now, the consensus is that several amino acids are known to play critical roles in this axis. For example, alanine, glutamine, and arginine, they stimulate alpha cell proliferation, while alanine, arginine, cysteine, and proline, they stimulate glucagon secretion from alpha cells. Most of these amino acids were increased by GCGR inhibition in our study and also reduced by PK activation. So we think these changes in these key amino acids are likely caused the increase or the reduction of alpha cell mass in this study.
So, Dr. Okamoto, switching to you, I hope that you can maybe provide us your view on the overall findings of the paper. especially in the context of emerging anti-obesity treatments that may modulate the glucagon receptor?
Sure. So one aspect of glucagon signaling we did not study in the current work is its lipid handling. To make glucose in the liver, glucagon promotes use of amino acids and lipids. Lipid breakdown in the liver in obese patients who often develop fatty liver will likely be beneficial. So here comes the idea of glucagon agonism for metabolic indication. But if you just fully activate glucagon receptor, that would lead to hyperglycemia. So there are many pharmaceutical companies combining glucagon and GLP-1, which by itself suppresses food intake and glucose. Now commonly found in clinical trials of these co-agonists and our mice with PKA activation are reductions in circulating amino acids. What we additionally observed in the mice are alpha cell atrophy and reduction in pancreas mass, which may be a result of low amino acids. So I would be gratified if developers of glucagon-based co-agonists can find our mouse findings useful as they evaluate safety profiles of the co-agonists.
Wonderful. Thank you.
Dr. Okamoto, are there any other key points or thoughts you'd like to leave us with as we close the interview?
Well, so the mechanism of alpha cell and pancreas mass reduction after liver PK activation is something, like to investigate next. Is it low amino acids causing both? What is the cell sensing mechanism? With the publication of this work, Kehan completed his postdoc fellowship and studied a new role at Regeneron. So I'd like to work with a new fellow to take on these questions.
That's awesome.
Well, Drs. Bao and Okamoto, we want to thank you for joining us today and for sharing your expertise and discussing your recent work on hepatic glucagon signaling and hepatic islet crosstalk. I encourage the listeners to read the full article in the June issue of Diabetes.
Thank you very much for having us.
Thank you.
The next paper we're going to discuss is one of our featured papers titled The Effect of Small Increases in Blood Glucose on Insulin Secretion and Endogenous Glucose Production in Humans. Today we are joined by Greg Kowalski, Associate Professor within both the School of Medicine and also the Institute for Physical Activity and Nutrition at the Faculty of Health, Deakin University in Victoria, Australia. Welcome, Dr. Kowalski. Congratulations on your paper, and thank you for being a part of this month's podcast.
Good morning, everyone from Australia. It's nice and early. Thank you very much for having me.
Now, Greg, if you've heard our podcast before, we like to start with a unscripted question, right? A little icebreaker. So since you're coming to us from Australia, I am curious, what is your favorite Australian animal? And bonus points if it's a known man killer.
My favorite Australian animal, maybe the drop bear, which we call koalas.
Oh, you
I
call them drop bears?
Well, they're koalas, but there's an old Australian kind of slang where they would call them drop bears, where a lot of foreigners would visit Australia and be like, "Oh, we heard you've got bears," and we say, "Yeah, they drop out of trees onto people." So
I like it.
Yeah, that's great. Greg, before we jump into the article, we love to give our authors the opportunity to highlight any other contributing authors or collaborators on this work.
So I'd like to thank all the contributing authors. This was a real team effort across both students, past and current staff members at Deakin University, and also our endocrinology colleagues at St. Vincent's Hospital and the University of Melbourne. So everyone worked hard to make this happen.
Excellent. Science is definitely a team sport. We've said multiple times on this podcast.
Yeah.
Can you start by giving us an overview of your research program and then maybe touch a little bit on how this project kind of stemmed from that program?
Sure. So over the years, much of my work has focused on understanding the mechanisms, driving whole body and organ-specific insulin resistance. And I'd probably want to say that firstly, I'm not a clinician, but rather a lab-based scientist. Originally during my PhD and early postdoctoral studies, I almost exclusively used rodent models, including transgenic animals, in vivo gene manipulation techniques with viruses, and a lot of mass spectrometry to study glucose and lipid metabolism, particularly in the setting of diet-induced obesity. However, upon moving to Deakin University to continue my postdoctoral studies, having gained access to great human research labs, as well as our own in-house mass spec, in our case, the GCMS, my research really quickly shifted to humans. And my then supervisor, Professor Clinton Bruce, who really had extensive experience performing invasive human metabolic studies and is actually the first author on this paper, we really began to design and conduct a range of basic human metabolism and endocrinology studies, particularly focusing on glucose, lipid, amino acid metabolism, and particularly really how insulin and glucagon control hepatic glucose production under a range of physiological settings. So I guess with this shift to doing human studies, our research somewhat naturally started to shift away from the question of what caused tissue-specific insulin resistance and rather move to what causes hyperinsulinemia and what is the difference between, for example, fasting versus postprandial hyperinsulinemia. And perhaps most importantly, like how do the pancreatic beta cells actually sense insulin resistance and therefore generate an appropriate degree of compensatory hyperinsulinemia to achieve normal blood glucose concentrations? So this is where this specific study comes in.
Yeah, that's great. Let's dig in a little bit deeper on the scientific premise for this particular study. And what was your hypothesis that you wanted to test going into this project here?
We wanted to really test whether very small changes in glucose could be potent signals, whether that was to the beta cells and or the liver. And the reason I guess we were thinking about this was that, I mean, it's well known that glucose is the most important signal that regulates insulin secretion. And we also, in turn, know that insulin is the most important hormone regulating glucose metabolism. So we found a sort of logical substrate for communicating metabolic demands. But I guess we were really puzzled by a few factors. One was the one I just described earlier. So in insulin resistant states, if glucose concentrations in the overnight fasted state are normal, then what drives and maintains compensatory hyperinsulinemia? We also started to notice from some of our own studies that we were conducting was that people, apparently healthy young adults, could have shifts in their fasting glucose concentration. So we would have participants visit us in the lab for a few consecutive weeks, and they had very stable fasting glucose. Let's say sometimes on the higher end of the normal fasting range, so say 5.5 millimolar. And then we would see them, for example, they would enter a study 1 or 2 years later, randomly. We'd see them again. And then we noticed that, hang on, now they're on the lower end of the normal fasting glucose range. So consistently for a few weeks straight, they were 4, 4.1, 4.2, 3.9, just bobbing around that lower range. And sometimes we saw the reverse. People started lower and then got a little bit higher. So it really made us think, I mean, is this meaningful? Is this got some kind of metabolic underpinning? And I guess independently of that observation, we were also really interested in classic studies from Professor Bob Wolf and Professor Gerald Reaven, real pioneers, where 30 to 40 years ago, they showed that even infusing very low rates of glucose into people, you would get very small changes, if any, in glucose and no apparent changes in insulin. But yet their endogenous glucose production would be suppressed robustly. And so together, this really just spiked our interest in, well, could these really tiny deviations in glucose be communicating a lot to the pancreas and or the liver? So this is really where this stems from.
Nice. Can you tell us a little bit about what you did to test this hypothesis then? Like who were your subjects? What was the sort of glucose manipulation strategy that you utilized here?
Sure. So we wanted to somewhat both recapitulate and expand on the older low-rate glucose infusion studies performed by Bob Wolf and colleagues more than 40 years ago. First, we, in 10 healthy young adults, 5 male, 5 female, after an overnight fast, we performed 2 separate trials in random order, roughly 1 to 2 weeks apart, where we then infused saline, which was our control condition, or glucose at a low rate via forearm venous catheter. We used the 1 milligram per kilogram per minute glucose infusion, which is exactly what Bob Wolf had done so many times all those years ago. Now, I thought probably it's good to give some context. I mean, why we call this a low-rate glucose infusion. Most adults have a basal glucose turnover rate of around 2 milligrams per kilogram per minute, and our infusion was half of that, 1 milligram per kg per minute. And in absolute terms, if you think about, say, like an average adult weight, let's say 75 kilograms or maybe 165 pounds, this basal turnover rate equates to around 10 grams of glucose going into the circulation via endogenous glucose production and 10 grams of glucose leaving the circulation due to tissue glucose uptake and metabolism. Because we were infusing glucose at a rate that is half of this is why we called it a low rate of glucose infusion. And now further on, we also infused our participants with a stable isotope, glucose tracer, 6,6,2H glucose, so that we could, during the experiments, measure their endogenous glucose production rate, which is where the liver and the kidneys, to a smaller degree, produce glucose endogenously, and so that we could also measure tissue glucose uptake rates. And we also sampled blood very frequently, every 10 minutes. And in a separate set of experiments of our endocrinology colleagues at St. Vincent's Hospital, we repeated these experiments in 5 participants with long-standing type 1 diabetes under clamped basal insulin concentrations that were carefully infused and titrated at a dose that allowed the participants to be euglycemic. And so we did this in order to determine the metabolic response to low-rate glucose infusion under a scenario where there will be no endogenous insulin secretory response.
So, Greg, I want to start with your findings on first the healthy controls, right? So your infusion, your low rate infusion of glucose caused a small increase in glucose levels. What other variables changed with that small glucose increase? And how do you think that informs us about what we know about glucose homeostasis?
So indeed, in our participants without diabetes, the controls, within 10 minutes of commencing the low-rate glucose infusion, there was only a very small glucose increase, around 0.3 millimolar. And beyond this point, it did not change. So for the remaining 80 minutes, glucose stayed stable. Now, this indicated that already within 10 minutes, there was a new metabolic steady state created, and this was associated with a rapid but only small, approximately 20% increase in insulin secretion. Now, this insulin secretory response really did not change beyond the 10-minute time point, so it really paralleled that of glucose. And also, interestingly, there was also a small but gradual suppression of plasma glucagon and free fatty acid concentrations. But again, this was delayed and did not adhere to around 60 minutes. Now, our glucose isotope tracer data revealed that in parallel with the small rise in glucose and insulin, that within 10 minutes of commencing the infusion, endogenous glucose production was suppressed by around 40%, while whole body glucose uptake was unchanged. So this rapid suppression of EGP basically mimicked the exact amount of glucose we were infusing. So it's suppressed by the same amount that we infused. And that explained why glucose no longer continued to rise. So it was really quite profound effects, and they really recapitulated what Bob Wolf had found.
Yeah, it's really an elegant balance there.
Yeah.
So now in your patients with type 1 diabetes, now you can kind of look at the system when you don't have that endogenous insulin secretion. So what other differences did you see in this particular patient population?
So indeed, unlike what occurred in our control participants without diabetes, where a metabolic steady state was achieved within 10 minutes and glucose only increased by 0.3 millimolar on average, our participants with type 1 diabetes were really not able to achieve a metabolic steady state. So their blood glucose actually steadily and in a linear manner increased throughout the entire period of the low-rate glucose infusion, and in fact, it peaked around 2 millimolar above baseline. And by design, due to the constant exogenous basal insulin infusion that we performed, the insulin concentrations throughout that experiment did not change. They were at basal. But unlike in our control participants who had a small insulin secretory response, which produced a gradual suppression of glucagon and free fatty acids, in our participants with type 1 diabetes, who again had no insulin response, there was no change to glucagon or free fatty acids. But it did, however, appear there was a very transient and partial suppression of endogenous glucose production. So this was probably glucose mediated. However, this could not be sustained. And therefore, this explained why the gradual hyperglycemia developed in these participants, but not in the controls.
So I know we've kind of touched on this already, but I wanted to give you an opportunity to sort of talk about your work and how it fits into the broader base of literature and what we know about the regulation of endogenous glucose production.
Sure. So I guess there's probably a few things to say. So our results strongly suggest that in people without diabetes, very small, perhaps barely quantifiable increases in insulin secretion have potent suppressive actions on EGP, and also glucagon secretion in adipose tissue lipolysis as well. And through comparing the responses between the control participants and those with type 1 diabetes, it appears that at least in the post-absorptive fasting state, small changes in insulin secretion rather than changes in glucose concentration appear to play the most important role in suppressing glucagon secretion. And furthermore, it also appears that under overnight fasted conditions, a small rise in insulin is far more important for suppressing EGP than is a small drop in glucagon. And I guess how does this fit in the literature? I guess I'll try to keep it simple, but I would say that under physiologically overnight fasted conditions, where natural islet hormone secretion occurs, EGP, or endogenous glucose production, is far more sensitive and responsive to the suppressive actions of insulin than perhaps is commonly recognized. And this also kind of makes measuring hepatic insulin action actually very tricky, especially under post-absorptive fasting conditions, because it appears that it only takes really a minuscule change in glucose and insulin to induce a large change in endogenous glucose production. It can be very easy to miss very small, subtle yet very physiologically meaningful changes in hepatic insulin action.
Do you think that relates to, or how would you think about this in terms of the pre-diabetes to type 2 diabetes transition? Do you think this work has implications for understanding that transition, or maybe even, sort of that idealistic version of, can we predict who's going to be susceptible to diabetes?
I definitely think that this study probably provides some small confirmation of the hypothesis that, for example, Gordon Weir and colleagues from Harvard have had for a while, and others have sort of talked about this, but that maybe very, very small changes in glucose precede beta cell adaptation. I mean, they could only occur perhaps for a few hours or a few days or a few weeks, but in cross-sectional studies, we're probably missing them. So, maybe, yeah, glucose is the most important signal for communicating beta cell adaptation and perhaps also to controlling liver glucose metabolism. Now, whether you can predict it, it's probably very challenging because, again, we're talking about differences that appear to be almost unmeasurable. Like we were able to detect them because we sampled so frequently and had a saline control group. But in a sort of population setting, it'd be very easy to miss changes that are 0.1, 0.2, 0.3 millimolar for many reasons.
So, Greg, I guess I want to give you a chance. Are there any other key points or thoughts that you want to leave us with before we wrap up?
I guess I would say, like, it would be, this to prove this hypothesis that very, very small changes in glucose perhaps drive beta cell adaptation and compensatory hyperinsulinemia, it would take a lot of work. And I don't think, you would have to design some kind of really prospective long-term longitudinal studies that sample blood very frequently and measure a range of metabolic parameters. And now these studies are not particularly sexy, but they would take a lot of time and effort. But I think it's almost the only way to really work out and map the evolution of hyperinsulinemia. And also in reverse, map the reversal. So yeah, I think, these are hard studies and they're not particularly attractive, but I think this is the only way to really, in a human population, be able to work this biological conundrum out.
Yeah, very true.
Well, Dr. Kowalski, thank you so much for joining us today and sharing your expertise and discussing your recent work on small changes in glucose and endogenous glucose production in humans. I encourage the listeners to read the full article in the June issue of Diabetes.
Welcome to another edition of Sweet Talk. I'm Kevin Williams, and today we're joined by Dr. Shingo Kajimura, who is this year's recipient of the American Diabetes Association's Outstanding Scientific Achievement Award. I'm also joined by our co-hosts, Dr. Darlene Sandoval and Dr. Kirk Habegger. Dr. Kajimura is a professor at Beth Israel Deaconess Medical Center and a Howard Hughes investigator. His research on brown and beige fat has reshaped how we think about energy balance and metabolic health. Shingo, welcome to Sweet Talk.
Thank you. Thanks for having me.
So, Shingo, we are excited to celebrate your career today, look back, and also look forward on the role of adipose tissue biology in health and disease. But before we get into that, we typically start off with a little bit of an icebreaker. And so the question we have for you is Is there anything outside of the lab, any hobbies or interests that you might enjoy and that might surprise some people?
Yeah, I don't know if this surprise people or not, but I love fishing. Actually, you see this, the fishing is my hobby, and actually the original curiosity about biology is it really come from fishing. I was born and raised in suburb of Tokyo, waterfront, and in my job per chore is to catch fish for dinner. So
Nice.
and I still think that this is, it's one of my hobby, and then I even think that this is my career plan B.
Perfect.
All right. Well, congratulations on being this year's recipients of ADA's Outstanding Scientific Achievement Award. That's just so cool. The award is definitely a significant milestone. Tell us a little bit about what it means to you personally and professionally to receive this award.
Yes, first of all, it's an incredible honor, and I'm humbled. But it just like unexpected in a way that a little different from maybe other recipients in the past that I didn't start my scientific career try to understand or cure diabetes. I really started with a fundamental understanding of sort of environmental adaptation. Just related to what I said about fishing, my biological curiosity, really the curiosity is about how fish adapt to different environment. For example, some fish can live in the muddy water, some fish can adapt to fresh water or seawater. Really, I've been really curious about how animal can adapt to different environment. And then I did my part of my PhD in Michigan, where winter is pretty brutal, and then I start to think how animal can adapt to cold environment for prolonged time, and that's what I started brown fat research without thinking anything about metabolism or type 2 diabetes. But for the last 12, 13 years, science turned out to be more relevant to metabolic disease and where I am, but I never started by questioning diabetes. So a little different.
Wanted to kind of have you maybe expand just a little bit more on your background, right? Your group really focuses on brown and beige fat. So for those who may not be quite as familiar, can you maybe walk us through what makes adipocytes brown or beige, or what do we really mean there, right?
Yeah, so the brown fat, the color actually come from mitochondrial iron, right? Brown fat is known for called heat organ in originally in hibernating animals, and that's what I got started interested in. But it's also the cell type that contain mitochondria at the highest level in the mammalian cells. That's why the brown. And then white fat, on the other hand, have really little amounts of mitochondria, energy storage, and it's been well known. So what I got initially interested in was maybe brown fat as a unique model to understand mitochondria, because you can compare what makes the difference between brown fat and the beige fat. Okay, brown fat is a embryonically defined thermogenic organ because newborn babies don't have enough muscle to shiver, so they need heat organ to keep body temperature without muscle shivering, right? Now, along line, we and others found that there's another called recruitable form of brown fat. Eventually, Bruce Spiegelman's lab, when I did my postdoc, we start to call them beige because the color is little less brown compared to embryonic form of brown fat, but that white fat can adapt thermogenic phenotype. And essentially, a recruitable form of thermogenic fat that exists in adults. And that's what really is important in adult humans. This is sort of, developmentally, they are very different. Regulation is different, highly inducible, and then also functionally a little different. So this is sort of the transformation that we learned for the last 10 plus years.
Yeah, thanks.
That's a perfect background of this field. And, you've contributed to a number of really foundational studies, discoveries, such as the identification of PRDM16 as the master regulator of brown and beige fat, as well as some UCP1 independent ways that brown and beige fat can regulate metabolism. So I wonder if you could tell us a little bit more, build upon what you just went through with that background of these stories. How does this, the identification of these mechanisms help us understand adipose tissue function, development, and maybe in health and disease?
I see. So one of the questions at the time when I started my lab, my postdoctoral fellowship in Bruce Spiegelman's lab was what makes brown fat different, right? Why both brown and white fat are, both are fat cells, but functionally very different. And the mitochondrial amount is completely very different. So why they're different? So that's a really fundamental question. And then together with Patrick Seal, he's now Professor UPenn, and then I really tried to understand the transcriptional regulation of brown fat. The why we focus on transcriptional factor is because this is about tool discovery, right? If we understand or discover master regulator, now we have a way to manipulate the pathway so that we can understand the function by knocking out or gain of function, so we can really establish causal relationship. So this is historically, scientifically, probably influenced by Bruce Spiegelman, who discovered PPR gamma as the master regulator of fat cells. So the discovery, well, why that is important was because now scientists can manipulate fat cells by targeting PPR gamma, right? And so that the master regulator upstream. So same analogy, if we can identify the master transcriptional factor, then we can make more brown fat or ablate brown fat. So that's the motivation is. So then we discovered it's called PRDM16 complex. When we start my own lab at the UC San Francisco, we wanted to understand how this molecule works. And so we did a lot of sort of purification of the complex, how each subunit works, very fundamental, without thinking any therapeutic application, honestly. But once we find the master sort of subunits or particular genes that are required and also sufficient, then we can make mouse models and we can make more beige fat, or we can make brown fat ablation mouse. That's what the discovery is. So when we start my lab, then we found this particular sort of core subunits of the PRDM16 complex. And then this actually allow us to develop the first mouse model that lack brown and beige fat without using diphtheria toxin. Back in the old days, Jeff Flyer's lab made beautiful work using diphtheria toxin to ablate brown fat, but that has a lot of confounding issues because of inflammation, right? So we made this brown fat ablation mouse, and then without thinking much about therapeutic implication, or we just initially thought this is just a cold sensitive mice. That's what we wanted to see. It turns out that this mouse developed severe insulin resistance, and then independent of body weight, and a phenotype was puzzling to me and surprising to us. And then, but immediately told us that the role of brown beige fat is more than making heat. They have play a role in glucose homeostasis, insulin sensitivity, and that's what the lab started to get into the field of type 2 diabetes.
So that bridges really nicely to my next question, which is focused on something you've been able to do, which is kind of bridge the basic science to looking for translational opportunities. Are there any particular strategies that you utilize to try to figure out which targets might be more translatable to sort of move forward? Or
Well, it's truth be, I don't. I don't have any magic ball in front of me to see this is gonna be the great target.
yeah.
But that's actually true for historically. That's what it is, right? I always try to answer the next important question, and then let me tell you a little bit about the discovery of UCP independent pathway. It turned out to be, I think, to answer your questions, and probably that's very important target for body weight maintenance. I'm going to talk a little bit later. So because of the puzzling observation we made, because brown fat ablation mouse have insulin resistance, suggesting that they're doing something more than making heat. And then we did a very opposite experiment, gain function experiment, and that eventually provided direct evidence that of UCP independent pathway. UCP was thought to be the only thermogenic protein in mammals, and in field of brown fat for over 30 plus years, including myself, believe that this is the factor that makes brown fat unique, and therefore making more UCP1 is a good thing as a metabolic health. It turned out to be not the case. UCP1 is not the only factor. Still, I think is important, but there are far more depth in that area. My personal thing is that we're still scratching the surface of it. UCP independent pathway is very important for we call metabolic health, glucose homeostasis, and probably called metabolic efficiency that we related to overall energy expenditure. So we can do is at this moment is to really try to better understand how that pathway really works, and hopefully that leads to the target. The other way to approach this is often combined with disease state, or let's say efficacy to certain medications. Probably everyone think about GLP-1 nowadays, but so that way we can sort of understand not only from basic science standpoint, but how that matter in pathophysiology, how it's regulated or dysregulated. This may often tell you the rate limiting step.
Yeah.
So I want to take a little bit of a left turn here, literally. You've had a remarkable international journey. And from your early days in Tokyo to some training in Hawaii, a position at UCSF, and then Harvard, and now at Beth Israel. Can you share a bit about your journey and how your early scientific interests shaped your career path? And I guess what I'm really driving at here is following maybe passion rather than the obvious logical step forward, right?
Yes, I retrospectively, I have zero idea how that happened. Really, I never even intended to. So I left Japan. I moved to Hawaii as part of my graduate school, and then studying fish physiology, and then moved to. I did my PhD in Michigan. That time I had no vision in terms of how my career looks like, and then I wasn't even sure if I could stay as academic scientist. But I incredibly fortunate. Probably one of the really the life-changing moment was like was very fortunate to work with Bruce Spiegelman. He's my mentor, friend, but really influenced my thinking. And then I met a lot of great colleagues and friends, and then really tried to address the next question, and then getting more and more into the field of metabolism. And then now I'm here at the Division of Endocrinology, Diabetes, and Metabolism. How can it happen? Right? So I really follow where science tell us to go rather than projecting or limiting myself to where I'm, so it's really random, serendipical, but all I can say is that I've been lucky, very lucky.
I suspect there's a little more to it than that, but I love the honesty, because I think for people coming up, especially a graduate student, they see us in our academic careers, and it just feels, a lot of what I hear is it's sort of this black box of how do you get to that point. So I'm just curious. I mean, you've had a lot of trainees across the years. Is there any particular advice you'd like to provide to young scientists?
Well, first of all, I consider myself as young, and then I'm still try to learn something new, and then
You're
try.
older than the younger scientists. How about that?
So, but the retrospectively, what really drives me is the curiosity, and also try to be a diver to go down to the bottom of the question rather than being a surfer, right? So I pick a question and try to get to the point that nobody really cares. That's why my strategy is. So that's always fortunate to work with really talented fellows and students, and the conversation start with what you're interested in rather than I interested in, because what I can teach them how to think, how to solve the questions, or how to write papers, how to write grants. But the only thing I cannot teach is the curiosity and an internal drive and passion, right? So that I really ask, tell me your passion, tell me the question you want to ask. Then I'll tell you how to climb the mountain together. I inherited the strategy from Bruce, my mentor as well, that he always have a lot of questions. The discussions never be one direction. It's always some conversation, and then try to meet the mutual interest, but to try to work to tackle a question together.
I love that.
Yeah, I love the analogy of working together to climb that mountain. I love that. And Shingo, finally, what are some big questions that you're really interested in tackling going forward? What are some exciting questions? I don't know, maybe just questions in general in the field of diabetes that you find interesting and that could be a major focus for research going forward.
Yeah, I think, generally speaking, very vague, but I think of some two things I want to say. One is that as a metabolism, it's such a vague concept of what is metabolism, right? Historically, molecular biologies, try to understand based on one molecules how this works. Metabolism is always a sort of vague concept. How exactly? What really breakthrough in this field is the discovery of master regulator, for example, leptin, right? How fat cell talks to the brain, or PPR gamma by Bruce Spiegelman, how fat cell development works, and so that. But now with the tremendous technological advance, sequencing at the single cell resolution, computational power, and really metabolism is to me, is the ultimate systems biology. I think this is a tremendous prime time to, and then I think we will see many discoveries for next couple of years. Really, that we built the tools that are getting ready, and then a lot of discoveries. That's one, more vague one. My personal mission or interest in our lab, particularly, is to understand what is energy expenditure. Right? Again, energy expenditure is how we use our energy. For the last 10 years or so, it's about we study brown fat as a way to understand adaptive thermogenesis, how our body adapt to cold or high fat diet or many external cues, adaptation. This accounts for probably 10 to 15% in humans, surprisingly low. What drives them, contribute to the most in adult human is called basal metabolic rate, which accounts for about 70%. We have very little molecular dictionary to describe what's the basal metabolic rate means. So what we hope to understand is build on this discovery of UCP-independent pathway. I believe that basal metabolic rate is UCP-independent, and we really try to come up with a molecular language that describes basal metabolic rate that accounts for 70% or more of energy expenditure in adult humans.
Great. Thank you for joining us today. Also, for our listeners, don't forget to join us at the ADA Scientific Sessions in June in Chicago, where you can hear Dr. Kajimura's Outstanding Scientific Achievement Award lecture in person. There's still time to register and attend this year's meeting. To register or learn more about the program, go to professional.diabetes.org/scientific-sessions. Thank you for tuning in to this episode of Sweet Talk. And as always, please like, follow, and subscribe for more diabetes bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.